Welcome back. We are here and ready for our bioinformatics wrap up. This is your time to shine. I see, just so that you know, for you to be able to share with us, we need your cameras to be on. So I see only a handful of schools have their cameras on. So if you want to share, make sure that camera's on so we can spotlight you. But we're going to let Sarah start the wrap up, and then we're going to look to everybody else to share with us. So I see a couple cameras. Can't wait to see. If you want to share with us, just raise your hand and I will spotlight you for the group. Sarah, you can take it away.
Yeah, thanks, Monica. Everyone, welcome back. I hope you had a fun time going through the databases. I hope Blast wasn't too slow today. All right. So we're going to start the bioinformatics wrap up. And the first thing I want to just jump into is let you guys kind of give your feedback on the lab. What did you learn? What is something that you found interesting? Did you get, how did you feel doing it? Did Blast take 20 minutes and you got kind of annoyed, so you played on Dr. No? Thoughts? And anyone
And
can feel free to unmute.
please unmute. Teachers, get to the keyboards to chat if you don't have your camera on. But if you have your camera on and you have the capability to unmute, we'd love to hear from you. Please do not be shy. I don't want to call on some groups that I see their cameras are on, but
Or if anyone
but
has questions they want to ask me about the lab before we start into applications of bioinformatics and other bioengineering principles and stuff that I do in the lab all the time, um, feel free to ask.
all right. While we wait for teachers to get to the keyboards and for students to get brave to share with us, Sarah, if you want to move on to your next slide, and then we can come back to the discussion once they're all ready to roll.
All right, so you guys can think through these questions for a little bit, and maybe the presentation will also give you some ideas on questions to answer. So the first thing I kind of wanted to start with was a more in-depth background on who I am. I gave you a very brief summary earlier, but today I want to talk more about my research interests, how I got to where I am today, and how my research that I'm doing right now is related to bioinformatics. And you guys can ask me questions about what it's like to do biological research at MIT, what it's like to do it in an industry setting, etc. So this has been my timeline from 2016, 2023. It's been a long time. And I started right where you guys are right now. I started as a high schooler doing my own, what I like to call freelance science kind of research, where I was basically just asking questions that I thought were interesting and developing ways to test them. So I would grow plants in my basement, or I would grow like whatever, make a makeshift lab in the back of our biology classroom. And this was generally just to tackle problems that I saw right in front of me. So one example is the pond near my grandmother's house. It was a pond that I had been walking around ever since I was a kid. And one day we went there and we realized that the whole pond was covered in algae, and all the fish had been suffocated as a result of this algal growth because the plants were actually sucking all of the oxygen out of the water. So that was when me and my friends kind of thought to ourselves, is there a way that we can make a sustainable mechanism for eradicating these algal blooms in my grandmother's pond, using low-cost resources and resources that aren't going to further damage the ecosystem within that body of water? And that was like where our first kind of science research project came from. It wasn't anything super sophisticated. It was really just stuff we were doing in our high school biology classroom. So then I got to MIT in 2020. I was a freshman during the pandemic, and I was really interested in bacteria. I'd done this work on algae. I wanted to figure out, I wanted to learn more about how bacteria work, how they talk to each other to exchange DNA, because it turns out bacteria have this really nifty characteristic where they can kind of touch each other and actually exchange DNA with one another. That's like if I were to shake hands with someone who has blonde hair and they were to pass me the trait for blonde hair, and I would instantaneously develop blonde hair on the spot. So that's pretty cool. And it also allows for bacteria to gain resistance to the antibiotics that we use to treat them, which is a really big problem in the coming years. So, I'd gotten my fill of fundamental biology research. I learned a lot about genetics. I learned a lot about genes. I learned a lot about bioinformatics. And then I wanted to do something more translational, something that was closer to the clinic, something that was closer to patients. So I ended up doing an internship at a company that some of you might have heard of called CRISPR Therapeutics. CRISPR is a gene editing technology, and there I was working within the regenerative medicine department to develop therapies for complex diseases like cancer and diabetes. And our keynote speaker, who you'll hear from in 24 minutes, is going to talk more about the really amazing work that CRISPR and gene editing is having on human health right now. Then I kind of switched gears entirely. I went to a more of a business sphere. I started healthcare companies in women's health. That's what got me really interested in applying my research to improve gender-based health disparities around the world. And that's kind of where I am now. I focus on gynecological cancer. I focus on gynecological disease, and I'm developing tools for early cancer detection. So the moral of this story is that you don't have to have any idea of where you're going when you start out. I had no idea where I was going when I was starting out, but I started out with just asking questions and investigating those questions with open source website resources. So if you have anything that you want to do in high school, you find anything that you're interested in, if you're interested in bioinformatics, or you're interested in physics, you're interested in astronomy, I recommend just going online and reading all about it, reading videos, sending emails to professors at universities. If anyone wants to email someone at MIT, I'm here. You can send me an email and I can try to connect you. So definitely just put yourself out there and see what you like to do. It takes a long time, but I think over time you figure out your niche. So some pictures associated with these so that you guys can see how janky my freelance research really was. This was the first lab that I worked in my high school. It was a lab that my friends and I created using bins that we bought from Target on sale. They were straight up $5 each. We cut holes in them and we grew plants inside of them to measure various things. So it was really not a sophisticated research project at all. It didn't require a lot of resources, but it did require a lot of ingenuity and a lot of thinking on our feet. So then here's all my beautiful algae. This was in the back of our biology classroom. We bought a little bit of algae online for like $10, and then we cultured it and expanded it again in my basement lab with those bins that you see on the left. And we created all of these cell culture flasks. And we did all of our experiments at the end of our classes. So I would finish with AP Bio for the day, and I would come to the back of the classroom and feed my algae. It was a very fun time. Came to MIT, I went to this much more fancy biological facility where I was actually doing real life sciences research with all of the equipment, with all the pipettes, with all the really intelligent people. And that's when I learned that I really do enjoy the research process. And now here's some pictures from where I work now. It's my picture from when I was at CRISPR Therapeutics. And here are some pretty microscopic pictures of some cells that we took a few months ago. So if anyone finds this picture and they think it's interesting, I would recommend looking into microscopy and looking into biological research. So now I wanted to give you a brief kind of overview of what I was doing during this research project where I was looking at early cancer detection. I'm going to tell you about it because it has direct relevance to what you guys are learning today in bioinformatics. So as some of you may know, conventional methods for cancer detection are really invasive and honestly ineffective. So there's really two methods that we use typically to diagnose cancer. First, we use something that's called a biopsy. So this is basically a surgical procedure, a mild surgical procedure, in which we extract a portion of the cancer cells in our body. And then we take those cancer cells, we expand them under these culture conditions, we look at them under a microscope, we put them through some sequencing, and we figure out whether they are actually cancer cells or whether they are healthy cells. Now, this has it has a few big problems. The first problem is that you actually have to open up the patient surgically to get this procedure. That's painful for the patient. But that's also has a chance of aggravating the cancer. If you touch the cancer in a way it doesn't like, it might start to spread and it might make it harder for us to treat it later on. The next way that you guys will probably see are things like CT scans, MRIs, and this is where we can actually get a look at what's going on in our body and see if cells are proliferating or becoming cancerous at a rate that is just not healthy. The problem with this one is that though it works very well, we can really only diagnose cancers that are solid, that are big, and cancers that we can see. So let's say you have really, really early stage brain cancer to the point where it's such a small dot that we can't detect it on a CT scan. Then how are you supposed to tell that patient that they have cancer? Because really, if we can detect cancer early, we can treat it a lot better and survival rates improve drastically. So the question that our lab asked ourselves, well, what about a blood test? I'm sure all of you guys have gone to the doctor for your annual physical and they have done some kind of blood test to figure out if everything is okay. And if they find out that something's not okay, then they do some more tests to figure out what the problem is so that they can treat it before it gets really bad. Well, why don't we do the same thing for cancer? What if you would go to the doctor for your annual physical and they would just test you for every cancer in the book? And that way, if we were to catch it early, you'd have a much better chance of treating it. So it turns out you can actually do this. It definitely requires more work. But it turns out that tumor cells in the body, that's represented by this blob on the left, they produce what are called circulating tumor DNA cells, represented by these little DNA strands. So every cell in our body just randomly secretes different forms of DNA, cell-free DNA, that are just floating around in our bloodstream. We don't exactly know what the biological significance of this floating DNA is right now, but the important thing is that it gives us a molecule that we can test for in the blood. So if the blood has a presence of these ctDNA, these circulating tumor DNA, then we can say, oh, maybe this patient has early stage cancer. So imagine you go to the doctor, you take a blood test, they have this tube, you put this tube through processing, and they find all of this DNA. The big problem is that the DNA is not actually colored differently in this way. There's not a color distinguishing whether this is normal cell-free DNA coming from a normal cell or whether this is ctDNA that's coming from a tumor cell. The only way to do that is through DNA sequencing, and it's through bioinformatics. So we have these two DNAs. We'll say the top one, the red and blue, well, let's say that is the circulating tumor DNA, and the bottom is just normal cell-free DNA. What we can do is we can take these DNA molecules and put them through some sort of sequencing technology. This is a DNA sequencer, and it can tell us where the mutations are, what the differences between those two sequences are. And people have done this for many different kinds of circulating tumor DNA and cell-free DNA. And through bioinformatics principles and other data analysis, we have been able to figure out what is the difference between these two DNA molecules and how we can detect it. So that's basically all my research. The other problem that we have now is that the body really doesn't like it when these random pieces of DNA are floating around. I think you can imagine DNA is a very important biological molecule. It has a lot of different functions. So the body naturally wants to protect that biological molecule. It wants to make sure that there aren't just random instructions floating around in the body. So it has incorporated immune cells whose job it is to basically eat these extra cell-free DNAs, these circulating tumor DNAs. And when that happens, the level of this DNA in the body becomes really low. So low to the point that even if we have the best bioinformatics principles in the world, we can't detect it. So basically, our approach is suppressing those immune cells that eat the cell-free DNA so that the levels are allowed to grow within the body. So a good metaphor I like to give for this is imagine you and I are out to lunch in some restaurant, and I know that you're really, really hungry. So I order you an extra sandwich and you eat both sandwiches, and now you're so full that you don't even think about touching my sandwich. That's kind of what we're doing here. We are feeding the immune cells a ton of different nanoparticles that make them full so that they don't take up the cell-free DNA. So the point of this story is that we really need a combination of bioinformatics principles with these biological engineering or just biological principles to create a real therapy that can go from the bench to the bedside. And then fun story, the work that I'm doing right now is actually being spun out into a company. So within the next year, there's going to be a company around the Boston area, and it's actually going to be commercializing these blood tests. So maybe in the next 10, 20 years, it'll be possible that you and I can go to the doctor and get a blood test for every cancer in the book. All right. So let's move on to some other applications of bioinformatics. The first thing is basically what we were just talking about, which is diagnostics. Those circulating tumor DNA cells are what we call biomarkers. They are biological markers that can be objectively measured, and they can be a sign of normal or abnormal processes. So in our case, that circulating tumor DNA is a biomarker that if we detect it, we can say that there is a risk for cancer. And the mutated proteins belonging to protein X and protein Y that you just identified as being associated with these diseases can be treated as biomarkers for these diseases. And we can use those markers as ways to diagnose these very complicated diseases. So these markers can be used to screen for the disease through blood, through urine, through tissue samples, enabling a potentially simpler and less invasive method for diagnosing the disease. And together, they can do a lot of things. They can help us predict how aggressive a cancer might be. It can predict how well a patient will respond to different kinds of treatments, or it can just identify early stage cancer altogether, which increases survival rates. The role of a biomarker that we investigated here is data analysis. So bioinformatics fits in right in this space, and then also a little bit in biomarker identification. You're basically throughout this lab learning about this very important process that is involved in us creating tests for various diseases. A good example of this is a company called ChromaCode that recently developed these low-cost, sustainable, reliable COVID tests to avoid relying on large labs and hospitals that might take 2 or 3 days to get your blood test back. So in low, middle income contexts, we can deploy these really low cost methods of COVID detection that rely on bioinformatics and biological principles. And we can enhance performance in testing at a very, very low cost. Now, moving a little bit away from diagnostics as a application area for bioinformatics, there's also gene therapy, which Valentin Sluch, who is our guest speaker, is going to talk a little bit more about. Gene therapy is basically a technique that modifies a person's genes to treat or cure disease. Because there are these mutations within our body that are associated with varying levels of diseases, gene therapy can go right in there, correct the mutation, and make it so that the body is now producing the healthy proteins that can exert the function that they were made to exert. There are a lot of different kinds of gene therapy. There is replacing a disease-causing copy of a gene with a healthy gene. There is deactivating a disease-causing gene that is not functioning properly, or there is introducing a new or modified gene altogether to treat a disease. And one method of gene therapy that you've probably heard about a lot is CRISPR-Cas9. So I'm going to skip this video, but I recommend that you guys go online and look up this video. It's by Mayo Clinic called CRISPR Explained, and it goes through the process of how CRISPR works, and it also goes through some really fun applications of CRISPR itself.
Sarah, would you be able to place that link in the chat to the video just so I can copy it and include it in?
Yes, I can do that after I'm done presenting. I can get the link.
Fantastic. All right, awesome.
All right, so now Some quick other biological engineering fields. I want to leave some time for questions at the end. These are the fields that I went over a little bit at the end of the intro presentation, but I wanted to focus on them more today. So this is synthetic biology. This is basically redesigning organisms to have abilities that they didn't have before. And we redesign these organisms by identifying genes or and introducing mutations that make them create new proteins, which allows them to have functions that they don't natively have. So some examples of stuff that we're doing at MIT or stuff that we're doing around the world that involves synthetic biology. Recently, some microorganisms, so bacteria, have been harnessed for bioremediation to clean pollutants from our water. So some bacteria have been genetically modified to be able to extract arsenic from water, from drinking water, which is a big problem, especially in parts of South Asia. Some people have been able to genetically modify rice to produce beta carotene. It's a nutrient that's usually associated with carrots that prevents vitamin A deficiency. So you basically have more nutritious rice, which is important in especially underprivileged contexts around the world. And we also have engineered yeast to produce rose oil, and people use that to make luxury scents. So you can do lots of different things with synthetic biology, and researchers use bioinformatics here to identify the targets to modify. Okay, the last thing that I'll talk about is neuroinformatics for those of us who are interested in the workings of the brain. I'll be honest, I don't know much about the brain at all, but that's kind of the theme that you'll see with neurobiology in general. We don't know much about the brain. So maybe you guys will be the ones to be able to apply the bioinformatics principles that you use today to be able to figure out more about how our brain works and develop better treatments for devastating mental illnesses. And eventually, I hope that we'll be able to use these computational models and data analysis to better understand the brain structure and the function. So if you like today's, I definitely recommend you go on Khan Academy and look at his videos on molecular and cellular biology. So I will put in a link for this DNA sequencing video that I found from Khan Academy, or yes, DNA sequencing video I found from Khan Academy earlier today. Definitely check that out. It'll tell you how those sequences that you were analyzing are actually created. And if you enjoyed this, I think these are some other engineering fields that you might find interesting. So I will stop the share. We can share some of our findings today, and then we can also ask some questions. I can go in the chat and see as well.
I know that our students are here listening and learning. I don't know if there's any questions for Sarah before we move on to our next part of the agenda for the day, which is our keynote. I see Milton has popped on his camera. Hey, welcome, Milton. I know you were
Hey,
here. How you
Sarah,
doing?
I'm doing well. That was outstanding. Wow.
Yeah. Thank you,
Yes.
Betsy, for putting
Wow.
in the link.
Oh,
Yeah,
you're
Sheesh.
thank
so
you all.
welcome.
We also have, I think, there's a couple of questions in chat. So if you don't mind, Monica, I think can I ask? Yeah, can I
Go
answer a
for it.
few
Yeah, of course.
before we go to our amazing keynote speaker? All right. So the first one was, how did you conduct independent research in high school? Was it virtually? Was it in your classroom? Was it at home? And what did you do with it to leverage to colleges? Great question. I you saw the picture. I actually went to Target with my friends. I bought some plastic bins. I cut some holes in them, and I made my own research lab in my basement.
Sheesh.
And the way that you can kind of come up with these topics, it's kind of a problem solving, kind of trying to come up with a hypothesis is a long process. It's honestly longer than it takes to build a lab in my basement. And what it involves, what I recommend doing first off is going to the UN sustainability goals online and figuring out what are the biggest problems in the world. You look at those problems and you figure out, is there any of these problems that I have a personal connection to? Any of these problems that I am really excited about solving? And then on another piece of paper, I want you to write down all the things that you're really interested in. So for me, I wrote down stuff like plants. I wrote down stuff like biology. I wrote down stuff like creativity. I like drawing. I wrote down everything, like literally anything. And I figured out whether there were connections between the things that I liked to study and the problems that I wanted to solve. So giving you an example, cybersecurity is a big problem. but I don't think I can solve cybersecurity with plants. But algal blooms, that was something that I could do. And that's why I had this idea to kind of um, do this. So once you have the idea, which involves really just a lot of research, looking at what's already been done in the field, talking to people, informing it, it's resources are limited, but you can find ways to make limited resources work, like waiting for the Target bins to go on sale. And then what did I do to leverage to colleges? I think one of the things that colleges really look for is excitement about a topic. They look to see what passions you have and what kinds of things you've explored. And I think what I put in my college application was that I'd been doing research for a really long time. It wasn't research in a lab, but it was research nonetheless. And I loved the process of being able to ask questions and come up with my own solutions. How can we contact you for questions? I think Monica, I think everyone has your email. So
Yes.
if anybody has like a professor they want, I am pretty good with my email. So Monica, you can feel free to like forward people to me.
Fantastic.
I always love being able to leverage the connections here to let people learn more. Yeah, I think that's all the
All right.
questions. Does anyone
We
have any more?
I think we're
Or
almost at time for our amazing keynote. So I want to say thank you to Sarah and everybody who's here. Let's give Sarah a round of applause for an amazing introduction, an amazing wrap up. Thank you so much for sharing your experience and your passion, which has led you on this journey, which is just amazing to hear about. And Milton, I see you're ready there.
Yeah, thank you so much, Sarah. I mean, game-changing presentation. Sarah is very special to engineering tomorrow. Homegrown, future scientist, game changer in the medical field for sure. You can hear her passion for this. You know what I'm saying? And one thing I really love is when she developed this lab, it makes you an advocate for your own health. I mean, that's a game changer. You know, it's we all need to be the ones to ensure that we care just as much about our bodies and our health and value ourselves as anyone else. So I love that this lab puts some tools in our hands, right? That we can become advocates for ourselves and our health. So thank you so much, Sarah. Thank you, Kate. Thank you, Sinai. Outstanding job. Look at this beautiful image of women in STEM. I mean, you all are cooking in hot grease. So, and Sarah, she actually gave us the plug for our presenter today. As I'm shouting out Sarah, I also want to shout out some of you all today. I saw you all working hard. You're engineering it. You're getting those algorithms rolling. But I saw you all having a lot of fun as well. And that's again, that's an attribute to you, Sarah, and this really, really dope lab that you have developed. Why am I here? I'm here to introduce this speaker, this guest speaker for bioinformatics. So you are going to be, you're in for an absolute treat. This is Dr. Valentine Sluch. He is a cell biology scientist, the associate director of CRISPR. Sarah talked about that today. He has research experience in stem cells, Cas9, which Sarah talked about, genome editing, molecular biology, biochemistry. He has experience in cell therapy as well. Very, very passionate about stem cells and gene editing. And look at those serious degrees. The degree in BS in biochemistry from the Oklahoma State University and a PhD in biochemistry, cellular and molecular biology from Johns Hopkins. I attended Johns Hopkins as well, but I didn't get that degree for sure. Can we please give a big round of applause for Dr. Valentine Sluch, and I will turn it over to him.
Thank you so much, Milton. It's great to be here today. I hope that the presentation works. I'm about to share my screen here.
Yeah, we can see you in that awesome background. Very
Okay.
nice.
Let's just get this going. There we go. I think we can see this now, right? Can everyone see this?
Yeah, we can see it.
All right, great. Hello again. I'm really excited to talk to you guys about gene editing, and I'm glad that Sarah has already introduced it. And I was reading the questions that you guys put together, and I was blown away by your interest as well as like your level of understanding. I wasn't sure to what degree high schools teach this stuff. It wasn't something that I learned in high school for sure. So when I was putting to like my talk together about what I was going to talk about today, I didn't want it to make it too technical, but I also thought that I didn't want to make it too simple either. But I feel like we're going to hit it right where we need to. And I'm guessing that's already went over CRISPR, but let's just go over it again, just so we're on the same page. So CRISPR is, one of the questions was, what's a simple way to describe CRISPR? The simplest way, I think, is molecular scissors that you can use to cut in the DNA exactly where you want to. And this has been a technology that's been a while in the making. It's something that was first reported in the 80s, where people were studying bacteria and they were seeing these strange sequences and they didn't know what to make of them. And over time, people figured out what was this system, and it's actually an adaptive immune system. So what happens in nature, which is something that I don't think any of us really think about, is that bacteria get infected by viruses all the time. And they're always trying to fight viruses off. And one of the ways that they can do this is through CRISPR. When a bacterium gets infected by a virus, viral DNA goes into the cell, and the bacteria can chop this DNA up if they survive the infection. And then they will take that chopped up DNA and they will actually integrate it into their own bacterial genome as a sort of memory. So then, if they see the virus again, they will transcribe those memory DNA sequences, make RNA, and the RNA will guide a nuclease, a Cas9 nuclease, to that DNA sequence and cut it. So that the next time you see a virus that you've seen before, you will chop up that DNA before it can do any damage. And this is how CRISPR works for bacteria. But in 2012, people figure out the mechanism. And they figured out that they could tell Cas9, the nuclease, to go where they wanted it to go in the genome and cut a specific place. And then they figured out that it wasn't really specific just to bacteria. They could do it with other cell types too. So they moved on into testing it in cell lines as well as in vivo later on. And they could see that you could actually use it to cut the DNA in mammalian cells and human cells in a very specific and precise way. And ever since then, there's been a lot of development in this field. There's been very different things you can do with CRISPR, many of which we're actually not going to get into today. But it has been a hot area of development, and people have been making improvements to it, and they've been adding different bells and whistles to make it do different things that they want it to do, essentially engineering the system further to something that doesn't exist in nature to get it to do something completely different. And some of the things we can do now is rather than just cutting DNA, you can actually change a specific base to a different base in the DNA sequence. And you can also make some defined insertions rather than just knocking something out or deleting a sequence. And in 2020, Jennifer Doudna and Emmanuel Charpentier won the Nobel Prize for their discovery of the CRISPR system. And in 2023, now we're awaiting the potential first approval of the first CRISPR therapy. And this is something that my company does, CRISPR Therapeutics, and it's a treatment for sickle cell disease. So I wanted to go back a little bit further before CRISPR. One of the questions was, did I ever think that a system like CRISPR would be around when I was in high school? And the answer is absolutely not. I did not think about this stuff when I was in high school. But at the same time, those systems actually already existed because there's something called a zinc finger nuclease, and that was discovered in the 1990s. And there's something also called a TALEN, and that was created and were developed in 2010. And both of these systems are other molecular scissors that you could use to cut the DNA in a specific place. And what made them less popular than CRISPR-Cas9 today is basically how hard they are to engineer. So for a zinc finger nuclease, the basic alphabet of how to get it to cut in a specific place is really challenging and has to be developed empirically by doing experiments. For talents, we actually do know the code. We know how to tell a talent to go to a specific place. But it's harder to make a talent because you have to make a new protein each time. For CRISPR-Cas9, what really set it apart and really made it take off for gene editing is that the protein Cas9 stays the same each time. What guides CRISPR to a specific place in the genome is the guide RNA. And guide RNAs are much, much easier to make. And you could make as many different guide RNAs as you wanted. And this is what's shown here in this orange diagram here for the Cas9 diagram. So rather than having to remake a new protein each time, you just have to change the guide RNA. You basically had to change the coordinates of where you wanted Cas9 to go and tell it where to cut. And actually, since the discovery of CRISPR, we've almost had, well, I guess over 10 years now since their original publication. There have been new systems that have been found also in bacteria as well as other species, some that were actually found in snails not too long ago, that essentially do some of the same things. They use RNA to guide an enzyme, a nuclease, to a specific place and cut at that particular junction. But CRISPR-Cas9 is very robust, and it still can, I would say it's the gold standard that everything gets compared to. And it's something that continues to work really, really well. So much like those other systems, CRISPR-Cas9 will cut the DNA at a specific place, and it will cause what's called a double strand break. So both of the strands of the DNA molecule will be broken. And then what happens after that is really not so much a Cas9 driven event, it's what the cell does with that double strand break. And then the cell has different repair pathways for DNA. Whenever it encounters a DNA break, it wants to fix it because it can't continue to survive if it has broken DNA. And the most common pathway that cells will usually use to repair double strand break is called non-homologous end joining. Which essentially just means that the DNA will, the cells will try to stick the DNA back together. And sometimes you will hear or read that this pathway is supposed to be error prone. And so people consider this fork on the left here as something that is harder to control and something that will create a break in the DNA and will have a change in the code. It will lead to a mutation. And the pathway on the right, homology-directed repair, is something that people often refer to as high fidelity. It doesn't make many errors. And for this pathway, what you need donor DNA. You need some sort of template to give to the cell and say, whatever break you have here in the middle, this is what you need to put in here. Use this template to copy from to repair your DNA break because this is what you should have in your genome. And usually HDR, homology-directed repair, takes place in dividing cells. So it's not going to happen in something like a neuron, which are non-dividing cells. So that's important because when you're trying to create a new cell therapy or just doing an experiment, what happens after the double strand break is introduced is not really up to us, it's up to the cell. And so for homology-directed repair, this is something we can do for things like stem cells that I'm going to talk about later, but it's not something that we can really exploit for many therapies. But it is very powerful because you can insert any gene that you want. You can replace a sequence. You can change a base pair. You can repair a mutation that existed before. You can add completely new genes. But for non-homologous end joining, this is more of something that you want to turn off. It can get a little complicated, but essentially you're deleting a sequence, or sometimes the cell will actually insert some random sequence into the break as well. But in either case, it will lead to something called a frame shift, which means that usually we read DNA in a linear fashion. And so if you imagine a string of letters, that's how that's going to get read. But if you delete one or two of those bases, then that message gets scrambled because now it's not reading right. So basically the spelling really matters. And if you delete bases or letters in a particular way where they don't make a multiple of 3, which goes back to codons and protein translation. But essentially, if you do it that way, it will cause a frame shift, and the cells will make something that's completely non-functional, and it will be what's called a knockout. So when we're knocking out a gene, that's essentially what we're doing. We're making a break that gets frame shifted, and then rather than making that protein that was made before, it will make something completely nonsensical. Of course, gets a little more complicated when you think about it, because even though this is primarily something to be done to turn a gene off, you can think about it from a different perspective, such as, what if I turned off a repressor? Then if I turn off a repressor, then I'm actually turning something else on. So it's not always just, I'm just turning something off. And so basically in the lab setting, we can use this technology to do pretty much anything. We can change any mutation. You can delete whole chunks of chromosomes. You can add thousands of genes. Some of it gets pretty technically challenging, but still you can do it. And you can make cell lines, you can make model organisms, such as you can make a mouse carrying a mutation that you're interested in studying, or a DNA sequence that is from people or anything like that. But as a cell therapy or as a gene therapy, CRISPR is more limited because we can't do anything that we can in the lab in the patient. So the most challenging is, of course, doing something in vivo, which refers to doing something directly in a patient. And this is also limited not just by the fact that we can't necessarily control what the cells will do, right? Because we can't control what the DNA repair machinery is going to do with the DNA break. So you're mostly relying on turning something off in that paradigm. But at the same time, you're also limited by where can you even deliver the CRISPR machinery. So currently, we are pretty good at delivering stuff to the liver. And so when you're looking up clinical trials for gene editing with CRISPR or other systems, a lot of times the liver will come up, and it would be used for rare diseases more often than not, but that's not necessarily limited just to rare diseases. You can actually think about knocking something out that leads to cholesterol production. And so you can lower people's cholesterol, their bad cholesterol, by knocking out a gene in the liver that everybody has. So potentially this could be a therapy that millions of people take to lower their cholesterol. An additional way that you could use CRISPR as a therapeutic is called ex vivo. So this is where you get into cell therapy by taking cells either from a patient or from a donor and modifying them in a lab with CRISPR and then infusing them back into the patient. And this is how the sickle cell therapy from CRISPR Therapeutics works. And one of the things that we are really cautious about and are most concerned about when it comes to CRISPR is off targets. So you may have heard about this or read about it online or wherever, where they talk about CRISPR and things come up in terms of like, what is the risk there? What could happen? And then a lot of the times they talk about off targets. So when you think about CRISPR cutting, yeah, it's supposed to go to that particular place in the DNA, and it's only supposed to cut at that spot. But there are potentials for it to cut somewhere else. And this potential primarily comes from guide RNA design. So remember, guide RNAs are basically the coordinates that you tell the Cas9 to go to. And if this is the molecule of bases that Cas9 is looking for, you can see that there's some mutations in red that are basically other sequences that look like your guide RNA in the genome. And so depending on how many mismatches of these red bases you have, increases or decreases the chance that you would have a cutting event take place. So usually for something like this at the top, we would refer to this as a good guide RNA because you really have no places in the genome besides this target gene at the top where you could expect to have cutting. If you have 4 mismatches, that's not going to cut. There is a limit in terms of how many mismatches are tolerated and still getting a cutting event. And there are general rules for that, but it's also something that you have to empirically test and evaluate. And on the bottom here, this is a guide RNA that I made up just to show what a bad guide RNA would look like. And so I just typed in a bunch of T's and a few A's. And we see that this sequence does repeat one time in the genome, but then if you change this one letter, you have another place in the genome where you have the potential landing spot. Change another letter, you have another spot, another one, you have another spot. So basically, this would be a bad guide because it would most likely cut at everything that only has one mismatch listed here, and potentially would also cut in places where you have two mismatches as well. So this would be something that you would normally not want to use for anything that you were doing. But regardless, even when you have a bioinformatic computer prediction that tells you that I should really only have cutting here, we have other experiments that we can do with cells in a lab, as well as potentially in vivo to assess, does that really happen? Is that really true? Do you only have cutting at that one spot? And we can also use statistics to then give us a probability of like, what's the likelihood that something might, a cutting event would happen somewhere we didn't want it to happen. And as long as you also know where that other event is, depending on where that place is in the genome, it may be important, it might not be important. So that's also something that you can evaluate. So after, now that we've covered CRISPR a little bit, I wanted to cover a little bit more about my background and my journey to CRISPR and like to being a scientist. And I was born in Russia and I moved to the United States when I was young. And my family moved around a lot. And eventually we settled in Oklahoma. And I went to high school in Oklahoma. And as I was thinking about my career trajectory and the choices that I've made, the thing that stood out to me is that I never really knew what I wanted to do for sure. I always had maybe some feelings about what it is that I liked and didn't like, but I never really felt like I was on a particular path. And even though like when I look at it laid out here, like it seems very linear and it seems like I went through it and I knew exactly what I was doing. But it really wasn't like that when I was making my choices. So in high school, I liked science, but I don't remember liking biology class more than any other science class. As a matter of fact, I think I like chemistry the best. And physics was also something that I liked, but not biology. And I took AP chemistry and I took AP physics and I took a lot of AP math courses, but I actually didn't take AP biology. And the main reason for that, when I was trying to remember why, was that I had a scheduling conflict because I was on a tennis team, and I either got to play tennis or I took AP bio, and I preferred tennis, so I stayed on the tennis team. And after high school, I went to college, and I didn't know what I was going to do. I didn't know what careers were really available. All I knew is that I kind of like medicine, and being a doctor seemed like a good job. So I was pre-med, and then I declared for a biochemistry major because it was related to medicine and it could help me get into med school. And that was basically what I thought I was going to do. But my brother, my older brother, he's only a few years older than me, and we overlapped in college, and he worked in a lab when he was in college. He suggested that I should go work in a research lab as well. And he said, it will look good on your med school resume, and like you'll get research experience, and it'll be great. And I didn't really know, how do you go and work in a research lab? And he said, just go and talk to professors. You're gonna work for free, so they're not gonna say no. So I talked to I think like 5 professors and settled on one lab where I started working as an unpaid intern. And they basically just had me do busy work. So it really wasn't stimulating. I wasn't really learning anything. So then I talked to another professor who was, he had a smaller lab and he was more open to me doing some actual experiments and learning more and actually doing research work. So I started working there and I worked there for a semester. And I was basically living in the lab, like all the time that I had, all the free time that I had, like after classes, I would just go to the lab and I would work on some experiments. And I had oversight. It's not like I was just doing whatever I wanted, but I was learning basic biochemistry, molecular biology techniques. I got to manipulate DNA. I was transfecting bacteria with DNA plasmids to get them to make a specific protein. And it was something that I found really fun. And after doing that for a semester, the professor that I was working with said, "How would you like to do this for a job?" So he started paying me, and I actually worked there in the summer, and then I kept working there throughout my undergrad. And it was it was funny because as I'm really enjoying working in the lab and being like a young scientist, my brother, who was working in the lab before and was an accomplished scientist, actually switched his direction and he decided he wanted to go to med school. So he ended up going to med school and now he's a doctor. But I decided that med school wasn't for me, and so I decided that I wanted to be a scientist. And so we switched our interests. And I was finishing college again. I was like, I don't really know what you do after this. I was going to have a biochemistry bachelor's. There's not too many jobs in Oklahoma that you could do with that. And so my professor in undergrad that I worked with suggested I go to graduate school. And I was not really thinking about graduate school at the time. I didn't really know much about it. And he really encouraged me to go and apply. And so I did, and I wasn't really sure what to expect. But most schools that I applied to were very positive, and they really liked that I had years of research already. That was something that they valued a lot. And so I got into multiple programs, but the one that I ultimately chose was the BCMB program at Johns Hopkins, which is an umbrella graduate program. And an umbrella program is something where it basically has a bunch of different fields that you could be in, but still be in this graduate program. And what that means is that you can pursue neuroscience, chemical biology, structural biology, genetics, whatever it is that you wanted, and it would still be under that same umbrella. So it's something that gave you a lot of options. And I'll tell you more about like my graduate school experience later. So just to quickly finish up this part of my talk, after graduate school, I again didn't really know what to do. Um, Normally you would probably pursue an academic position. So you would try to become a professor. And instead, I decided to do a postdoc in industry. So I went to Novartis, which is a big pharmaceutical company. And I wanted to do a postdoc there because I essentially wanted to get an experience in what working for a company in industry was like, and what was it like to try to find new drugs. And after doing that, I decided that what I really wanted to do was to use gene editing and stem cells as a medicine, and a position like that opened up at CRISPR Therapeutics, and I've been there ever since. So getting back to grad school for a bit, when I was going through that decision and deciding what I wanted to do after college, a lot of my experience in college in research was in structural biology. And so I thought that I would most likely end up joining a structural biology lab because that's what I had experience in. That's something that I thought I could just jump into and do very confidently. And then during my first year in graduate school, as I've been going through rotations and picking different labs, my grandfather that I was very close to got diagnosed with glaucoma, and it was not going well, and he was losing his vision. And it was something that really affected me because he was someone I really looked up to. And he was someone who was always busy. He was really full of life. He was a mechanical engineer by training, and he had a lot of patents. He was always inventing stuff. He was always like also starting small businesses, and traveling. And obviously, like when you go blind, you can't do any of those things. So it was something that really affected me, and it made me want to do something about this. So I decided to join a lab that was actually studying glaucoma and was trying to find new treatments for this disease. And basically, glaucoma is an increase in intraocular pressure. It's the pressure in the eye. And it happens because you have drainage in your eye and these canals get blocked. And as they get blocked, the pressure increases and it pushes on the optic nerve, which is in the back of the eye here. And that optic nerve starts to die. And eventually you will start losing vision. And then you will mostly start losing vision from your periphery inside in. So what usually happens is that because this is painless, you don't feel it happening. You don't think about it. So even though you're losing vision, you often compensate for that vision loss by simply turning your head. So you might not see something to the left of your eyes in your periphery, and so you turn your head and then you don't realize anything's wrong. So by the time patients actually go to the ophthalmologist and they say that something's weird, I have vision loss, they've actually usually lost something like 50% of their vision. And once that vision is lost, you can't recover it because the optic nerve, as pretty much all neurons, they don't regenerate. So once it's gone, it's gone. And it's estimated that 3 million Americans have glaucoma, but only about half of them know it, simply because they don't recognize that they're losing vision. There's no cure for glaucoma either. So the best course of action is early detection. And then you can be put on a treatment plan that will lower the intraocular pressure to slow down the disease progression. But unfortunately, that wasn't working for my grandfather. So he was progressing rapidly. And by the time I was done with graduate school, he had gone completely blind. But when I started graduate school, basically what I wanted to do is make human retinal ganglion cells. And this is a diagram of a retina. And you have different cell types in the retina. And the ganglion cells are the ones that are actually here on the bottom. And they're the ones that will form a bundle and send a projection from the eye to the brain. So they're the ones that are actually sending the signal from the eye to the brain about the vision that you're seeing. And these are the cells that die in glaucoma. And I really wanted to find a way to make them because we wanted to make human cells to study human disease. Usually we use mice to study diseases in biology or some other model organisms. And this has been very powerful, but mice don't get human diseases. So a lot of the things that we find work in a mouse often fail in clinical trials because people are not mice. And so the idea was, can we make human cells because they would be more medically relevant to this condition? And the way that you could make human cells at the time, these were all like pretty new technologies, was to use human pluripotent stem cells to differentiate them to make human retinal ganglion cells. So the first pluripotent stem cells that were discovered were embryonic stem cells, and these cells do come from embryos, but they come from donated IVF generated embryos. They don't come from abortions. So usually when a couple will have IVF embryos, they're not going to implant all of them. So the ones that don't get implanted, they're often discarded and thrown away. But you can also donate them to science, and then they can be used to make embryonic stem cells. And these stem cells are pluripotent, meaning that they can become any other cell type in the body. And so in this diagram, they're just showing you that you can make heart cells, neurons, as well as liver cells from them. And they will be human cells and they will behave just like human cells. And then you could use them for drug screening purposes or potentially to study development. So you'll be studying human development rather than mouse development. And probably the most lofty goal has always been to use them for cell therapy. And the process of going from an undifferentiated stem cell to a differentiated cell is called differentiation. And essentially that is sort of like cooking. You're giving the cells different proteins or media or chemicals, essentially giving them different signaling cues to say, now become this. And it's something that usually requires a lot of time and a lot of testing. And to figure out a recipe for making a particular cell type is a very difficult thing. And so even though we can make a lot of different cell types from embryonic stem cells, we can't really make all the different cell types that are in the body just because we basically don't know how. But another source of pluripotent stem cells altogether is induced pluripotent stem cells. So this is something that was discovered as I was finishing high school. And essentially what Shinya Yamanaka discovered and got the Nobel Prize for is that you can take any cell in the body, and he was primarily working with fibroblasts of skin cells, and then you can overexpress stem cell genes in those fibroblasts, and they will reprogram to something called an induced pluripotent stem cell. And these iPSCs are essentially equivalent to embryonic stem cells. They can differentiate to any cell type in the body, just like an embryonic stem cell, but they don't have any of those ethical questions in terms of where they came from. And when this was discovered, this set off a huge amount of interest in the field, and it basically started a whole new field of reprogramming. And people were very excited about using these cells for personalized medicine. So any cell type that you had that was deficient or missing, theoretically, we could replace it and then put it back into you to treat your disease. And people thought we could be treating diabetes, blinding disorders, heart disease, and Parkinson's disease. But basically, everything was a lot more challenging than people thought in the beginning. But one of the things that I definitely want to call out about iPSCs is that they're, in my opinion, probably some of the most magical things in the world. Because if you take a skin cell from a 90-year-old person, you can reprogram it to an iPS cell, and it will actually lose all of the aging biomarkers that it had. It would become young again. Another thing that is really wild to think about is you can take a skin cell from a mouse, make it into an iPS cell, and then implant those iPS cells in a mouse in the uterus, and it will make a whole new mouse from that one skin cell. I just feel like that's probably one of the craziest things that exists in biology. So I always found that to be very interesting to work on. So in graduate school, this is what I was doing. I was figuring out how do you make retinal cells. And this is what the cells would look like as I was doing my experiments. So you start with this sparse culture of cells that over time they grow and they fill out the lab dish. And by studying gene expression, we could tell that by day 40, we had retinal ganglion cells in this dish, but there's no way to see them. So there was no way to isolate them. So that's where CRISPR came in. So CRISPR was discovered when I was doing my PhD work, and we decided to try it. We were probably one of the first labs to try it, actually. I think it was 2013 when we did our first experiments. And what we did is we cut in this brain3B gene, which is a gene that retinal ganglion cells express. And we added on a red protein sequence right after this brain3B gene, so that anytime brain3B was made, you would also have this red fluorescent protein made. And here is just a DNA gel of what that looks like. So this is the control, amplifying this gene region. And then after genome editing, we have inserted this red sequence. So now it runs larger on the gel. And now, like when we differentiate that cell line, we see that these red cells start to emerge around day 25, and then more of them are born as time goes on. And by day 40 is usually when they would reach their maximum production. And obviously this was very exciting the first time that I did this, because I would check these cells every day for months to see if they would turn on the expression of this marker. And I think this, this picture is really one of the most beautiful pictures for me from my PhD. And all of these are retinal ganglion cells that are born in a dish. So these are human cells, and they're trying to send essentially a bundle of axons to the brain. There's no brain here in this lab dish, so they're searching and they're looking for a place to connect to. But they are fully functional, and we use them in a lot of different ways after making these cells. And we started thinking about transplanting them for experiments, but a lot of the things that we did was actually drug screening to basically try to find new treatments for glaucoma to prevent these cells from dying. And using that red signal, you could use something called fluorescence activated cell sorting to sort their cells and purify them. So you would have these pure cultures of retinal ganglion cells, and then we use them to study their gene expression, and like I mentioned, testing drugs. And then towards the end of my PhD, we did do some transplantation experiments to see if we could regenerate the optic nerve. And the lab has since gone on to do more of these types of experiments. So switching gears again, wanted to introduce what I actually do at CRISPR, which is trying to build a type 1 diabetes cell therapy. So type 1 diabetes is an autoimmune disease that essentially destroys your insulin producing cells. So normally you have these beta cells in the pancreas that are producing insulin in response to glucose to basically tell your body to take up sugar. And in type 1 diabetes, these cells are destroyed by your own immune system. And once they're gone, you don't have any insulin production anymore. So those patients have to start taking insulin injections in order to control their blood glucose levels. And this is something that is difficult to do, and something you have to keep doing every day. And you would die if you didn't do this. And patients are at risk of going blind from type 1 diabetes. They're also at risk of kidney failure, and they potentially could die at night if they don't manage their insulin very well. This is especially hard for children, because this is something that young children do get. And then, it's very obviously hard on their parents as well to have to monitor them. And one of the cell therapies that has been developed for treating type 1 diabetes is called the Edmonton Protocol. It was created in Canada. And what they did was they took cadaver-derived pancreases, and they took out the islet cells from them, and they purified them to basically get the beta cells. And then they would transplant them back into the livers of type 1 diabetes patients. They would transplant them into the liver instead of the pancreas because it was easier to do surgically, and it was much safer that way. And for these cells, they really don't need to be in the pancreas in order to control your blood glucose. They could theoretically be anywhere. They just need to have a connection to your blood supply. But in this cell therapy, you do have to take immunosuppression. So even though this cell therapy has worked, and it works rather well. So here we see blood glucose excursions, basically deviations in your blood glucose, and they should really be pretty flat if everything is working well. But this is a type 1 diabetic before the transplant, and this is after. So we see that it's a dramatic difference where essentially this person is now completely healthy, and they don't have to take insulin injections anymore. But in order for this cell therapy to work, you do have to take immunosuppression. And immunosuppression carries its own risks. You potentially increase your chance of infection as well as cancer, and also some other side effects such as nausea and weight gain. And another reason that this cell therapy is hard to do is because there are not that many suitable cadaver-derived islets that you could use. So one of the things that people have been working on for years now is to use stem cells to differentiate them to islet beta cells and then put them back into people as a cell therapy to replace the beta cells they have lost. And this has really taken a giant leap forward in the last 2 years, where a company called Vertex showed that they could take stem cells, differentiate them to beta cells, transplant them into people, into their livers, just like in the Edmonton protocol. And then they were able to stop taking their insulin. They became insulin independent. And so far, it has worked for a few people, and the clinical trial is still ongoing. But in this clinical trial, they do still have to take immunosuppression drugs because these cells didn't come from you. They didn't come from a match. It's basically like getting an organ donor, an organ donated from someone you didn't know. So you have to take immunosuppressants to prevent rejection. And the thing that we want to do at CRISPR is essentially take this one step forward. So this is a cell therapy that we've been working on with a company called Biocyte, where the concept is very similar. We take stem cells and we differentiate them to islet cells, and then we put them in a little device that gets implanted under the skin. And blood vessels then grow into the device. And once the blood vessels are in there, the islet cells can sense the blood glucose in your blood, and they can secrete insulin as needed, so you can stop taking insulin injections. And where CRISPR gene editing comes in is that we take the stem cell line and we gene edit it to make it invisible to the immune system. So now that your immune system can't see it, you no longer have to take immunosuppressant drugs. So essentially, if this works, you don't have to take insulin or immunosuppressants, and then you can go on and live a perfectly normal life. And essentially, it would be a cure for type 1 diabetes. And this is in clinical trials now, and we continue to work on this cell therapy. I'm going to stop here. Thank you for your attention. Happy to take any questions, Milton, if we have any.
Yeah, that was that was really good. I mean, I don't know. Can we clap it up for Dr. Valentin solution? I mean, that was just really, really good. He literally talked about reversing aging and creating new organisms, which this is the stuff you see on TV, man. I mean, this is it's like unbelievable. I'm too excited. But personally, to be honest, I don't know if we talked about this. I was diagnosed with cutaneous T-cell lymphoma like a year ago, doctor. And so just to hear that you and other students that are here are interested in this technology to cure diseases is a game changer, right? It's just there's no conventional way to address diseases like the one I have. So it's this was unbelievable for me. I'm gonna dig into some questions. I'll share my screen. There are a lot of questions for you, a lot of great questions. So let me share my screen. I think you talked some about this, you know, what inspired you. I love the story you told about I don't know if it was your father, your grandfather who had glaucoma, and how you kind of shifted some, you know, to research that. But what was the impetus of your inspiration? Where did it really start?
I think I've always been interested in treating diseases. And so originally, I actually wanted to be a veterinarian because I really like animals. But then I I guess I shifted because I thought that it would be more impactful to study human medicine.
Yeah.
But as I was going through the basically the process of getting yourself ready to apply to med school, I felt like I enjoyed being in the lab more than I enjoyed interacting with people. And I felt like a doctor needs to be a people person, and they really need to like have good bedside manner and not, you know, basically always be thinking about experiments. And I felt like additionally that if you wanted to find new treatments for anything, you had to do it at the lab bench. It wasn't going to be in a doctor's office. But I would also say that it was really mentors. Basically, whoever believed in you, whoever encouraged you,
Wow.
really spoke volumes. So like when my undergrad professor was like, you should go to graduate school, you're good at this. I think like this would be good for you. That was huge for me. Like I didn't really think about it before. So I feel like a lot of the choices that we make, they're often based or guided by other people that believed in us. And so like your life can take many different trajectories. And so I think it's always good to ask people for advice and to don't close off yourself to things either. It's like be open-minded.
That's really loaded. That's really good. Something else that you talked about it warranted this next question. You said you didn't even take AP biology in high school, right? So there's many
Yeah.
paths to folks getting involved with this career path. So how can students begin getting involved in this career from an early stage?
Yeah, I definitely think that for some of these things, I think you could do them in high school classes. I heard that some high schools do CRISPR um, experiments in the lab, which I think is crazy. Not from a safety perspective, just blows my mind that like you can just do
Yeah,
that now. But definitely in college, there are always going to be research labs that you could rotate in or join, and if they're not, like you just maybe ask if you live near the university, just email people and ask. Don't be shy. Worst thing is they say no. Um, you can also intern at companies. Some companies do outreach to high schoolers and have summer programs. And so I definitely think that in college, it gets easier to get exposed to these things. But if you're really motivated in high school, you can start by essentially asking professors or companies.
that's good. I mean, utilizing, like you talked about, the mentors, other folks around you to help, grow towards your success. That's great. Here's another one. What are some examples of cell therapies that can be used to improve the outlooks of patients who live with diseases? Now, for my disease, I started to think or be told that it had gotten kind of bad, and I didn't know if the conventional therapies were going to work, so I started to look into stem cells, right?
Mhm.
So it's a great question. Like, what are some examples of cell therapies that could be used to improve an outlook, the patient's outlook?
Yeah, I mean, for cell therapies, so basically, like one of the other questions was what's the difference between a gene therapy and a cell therapy? And there's definitely overlap. As a broad definition, it depends on what it is that you're delivering to the patient. So if you're delivering cells, it's a cell therapy. If you're delivering genetic material, it's a gene therapy. But for cell therapies, I think that the type 1 diabetes therapy from stem cells is, I think, one of the best proof of concepts about how this works. But a much more broad and common one has been CAR T cells,
Mm,
which are T cells that have been engineered to go and kill cancer. So this is something that's actually already approved. So you can, if you have cancer, you potentially can get this treatment. And currently, they're taking T cells from patients and they're genetically modifying them to tell them to go and kill a specific cell type. And so this has been approved for certain blood cancers now, and it's been very successful, and that field continues to grow. For other cell therapies, there definitely are other indications that people are testing in clinical trials. But I still think that for many of them, it's still early to really claim that they work.
I'm glad you hit some of those. So some of the students also asked about joint regeneration and brain damage. Are those more those future technologies, or are they on the cusp?
For brain, I would definitely say future. That's one of the ones that is really hard to treat, and the main reason is that once you develop all your neurons, they have connections, they have formed, and then putting new cells in there, making them form new connections again is very difficult. The cells don't know what to do innately anymore. So that one's tough. For joint regeneration, I looked into it before I came here today. So like that's on the cusp. It's coming. It's been a long time coming. But I feel like for that one, and probably in the next 5 years, I think that that one's going to be a big one.
I hope so, because my basketball days have depleted with these joints. So I'm just hoping to get back on the court soon. 5 years, that's all I have to wait. I'm there. Um, here's another one for you. We've heard that adult stem cells are difficult to find and grow. This is a challenge I found out. I was questioning as well. It's just the difficulty, right? We getting access to this technology. So what are some options? This will be my last question. What are some options out there for people who didn't get that access?
I definitely wouldn't worry about it. From what um, is out there is that no adult stem cell therapies have been approved. They have been tried in a number of different conditions, usually for joint diseases, but a lot of other ones as well. And uh, I would say the jury's still out, but for the most part, clinical success has been lacking. Um, and so if you didn't bank your cells, don't worry about it. For what we would actually have in the future, most likely, is banks of stem cells from a whole bunch of different people that will be able to match to you. And that is something that they're doing in Japan, and something that for blood banks they do all over the world. And then in the future, I think with iPS cells, you would have basically iPS cell banks that could do a lot of the same function.
Wow. Well, we have to do this again. This was so good. I took some quick notes on some recipes for success. I used that word recipe just because you talked about recipes for stem cells.
Yeah.
What I learned from you is you don't have to be born with it. You just have to love it. And you talked about living it, living in the lab. And also that there are multiple paths to achieve your passion and purpose. This was so good. I got lost in time, and Monica had to remind me where we were. It was so good. I learned so much. Thank you so much. Can we clap it up for Dr. Sluch one more time for this outstanding presentation? It was so nice. So
Thank you
nice.
so
Learned too
much.
much.
Thank
Also,
you.
we want to highlight quickly that we have another special lab coming up, Machine Learning Day, December 19th. Keynote speaker, hopefully I pronounced this right, Adam Shear. So thank you all again. I'm going to hand it off to the outstanding host with the most, Monica, to take us to the next steps. Thank you again, Dr. Sluch, game changer.
Well, I want to say thank you. That was an amazing presentation. But these students who were with us all day doing bioinformatics today, look at them clapping it up at Cardinal Hayes. Thank you so much. We had a fantastic day. Thank you for staying with us. Do not forget to complete your exit tickets. Students and teachers, you both have exit tickets, and we appreciate you being here. Please sign up for our next event, which we have one before the machine learning, which is renewable energy as well as space, and then machine learning. So we have a few events coming up before the Christmas break. Tell your teachers to sign up, and we hope to see you there. Have a phenomenal day, everybody. Thanks so much.
How in the world did I forget about space?